You're good to go.
Could I share my PowerPoint quick, just because I have a
Yep.
slide for introduction and everything? Perfect. Can everyone see this, or?
Yep.
Everyone can see this? All right. Okay. I'm doing a talk on just an intro to developmental biology for everyone who doesn't know. I'll start with a bit about me. I'm a fourth-year undergraduate student in the honors CMMB program, so Cellular Molecular Microbial Biology at the University of Calgary. And currently, as a summer job, I'm working at the Alberta Children's Hospital Research Institute under my PI, Dr. Sarah Childs. And in the lab, I am currently working on studying vascular development in zebrafish. So is everyone able to see my cursor here? Yeah, okay, so this is an adult zebrafish. So normally we would study the embryos, but of course you need adults to make embryos. So so the overview of my talk is I'll start off with just a bit of developmental biology at a glance to sort of get you oriented. Then I'll move on to some basic cell biology principles to fill in some gaps that you may have had in the past. And then I'll move on to talking about model organisms. So those are our ways as biologists to study how development works inside a living, breathing organism. And then I'll move on finally to the core of the matter, developmental biology. We'll look at stages, genomic regulation, and some experiments we can look at to sort of probe how development occurs, how it works, and what we can discover from it. All right, so first section, developmental biology at a glance. So first, I'm just going to start by showing everyone the life cycle of a mouse. So as you all should know, mice are a very common laboratory animal that we do experiments with. I'll get into this later, but one of the reasons being is because they're mammals and humans are also mammals. So we're very similarly related at the cellular level. So I'll go over this in more detail, but this is the general life cycle. So we start with an adult. They produce the gametes, which are the egg and the sperm. Once those fuse, we get the process of cleavage to form what's known as a blastocyst. This will implant into the uterine wall, and then we'll get the actual process of embryonic development occurring and fetal development. And once the fetus is born, it grows to become an adult again. So developmental biology in general focuses on the cellular interactions and signals required to turn a single fertilized cell into an entire organism with billions or even trillions of cells. So that's a huge change. That's a big difference. One cell into trillions of cells. So that's why it's such a detailed field that has a lot to discover still, because it's so complex. Development obviously occurs through all life stages. So even after you're born, when you're a child, you grow into an adult. But many developmental biologists tend to study embryonic development. And that's just because of the very unique cellular interactions that occur during embryonic development that result in the formation of organs, organ systems, different tissues. And in the end, we can really look at those and understand how our bodies form and how our bodies are made at the genetic and cellular level. All right. So this is kind of a fun activity I kind of decided to put in here. If anyone wants to participate, go ahead. But if you don't, then I'm totally fine with just saying what each embryo is. But I'm going to show you a bunch of embryos. And I just want someone to guess what they are. So it's kind of just an icebreaker to show what these embryos look like, how they develop. And by the way, sorry if you're not comfortable with images of embryos, there's going to be quite a bit. These embryos usually, none of them will be human, of course. We don't do experiments on humans. They will all be animal models that people are certified to work with. So don't worry about seeing anything like that. These will all be animals. But to start with, can anyone guess what this embryo is? Just type it in the chat or yell it out.
This looks
What
like a
animal is
lizard.
this?
This
It
looks
does
like
look like a lizard, but they are all very similar looking when they begin. So
Yeah.
yes,
Khalid, you wanted to
Khalid
say?
says mouse. Yeah, he said in the chat. Everyone's saying mouse. And yes, this is a mouse embryo, 11 and a half days after fertilization. So in less than 2 weeks, you go from a single cell to this, which is quite amazing. I mean, this is under a microscope, but you can't even make out the cells because they're too small still. So this is, I'd say, millions of cells right here, from a single cell to millions in less than 2 weeks. And this is a mouse just 6 days later. That is crazy. Less than a week later, and you go from this to this. So this looks like a lizard. This looks like a mouse. So it's quite crazy the difference that it makes, just a bit of time. And that is why developmental biology is so interesting. Like, how does the embryo organize itself? How can this occur autonomously without any external signals? So this is the next one. This one's a little more obvious, I think. Does anyone know what this is?
Is it a bat? Is it a bat?
Yeah, this is a bat. This is a bat embryo. You can see its wings here. I'm not really familiar with bats. People don't study them all that much. I assume this is a time stage. So this one's younger and this one's older, because you can see the webbing in its wings is kind of expanding as it gets older. Its tail is also growing longer. Its toes are getting more defined. And I'm not sure what these are, but they look like they're getting larger on the head here. But yeah, that's a bat. Can anyone guess this one?
It's a chicken.
Chicken. Any other guesses?
No, chicken doesn't have
Yeah.
tails. It has a tail, kind of.
Mhm.
Is it a lizard?
Yeah, this is a turtle. This is a turtle. So it's kind of hard to see, but there's a bit of a shell here. So even in embryonic development, they start forming their shell, which is crazy. And you can see sort of the webbing in the limbs. But if you'll notice, most of the embryos have webbed hands or digits, because that's just part of the developmental process. I'll show you later how that kind of forms into the fingers. So yeah, this is turtle embryo. What is this one?
A cat? Is it?
Yeah, this is a cat embryo. Good guess. Yeah, this one's kind of hard to tell. It looks a little bit similar to the
God, you
very
guys are
first one.
instinctive biologists. I can make one right.
This one is pretty hard. Yeah, as you can see here, what I said before, you do see, this is the hind limb here. You can see some webbing in between the digits, but of course that gets removed in an adult cat. So they all do look very similar. They grow in this sort of curved shape. And of course, this would be embedded in the uterine wall while it's developing. So this has just been removed from that. Usually, I think these cat embryos at the animal sort of where they take in cats, I can't remember what they're called. When they take in strays, sometimes the cats are pregnant, and they'll sometimes when the cats are euthanized, they'll actually be pregnant, so they can get the embryos that way. I'm pretty sure no one actually studies with cats. No one actually breeds cats for the purpose of studying them. But so this embryo probably would be an aborted embryo from one of those animal places. All right, this one's much more obvious.
Elephant.
Yeah, this is an elephant. This is an elephant. I'm pretty sure this is the only image online of an elephant embryo. I've seen it one time and it was the same image. So I don't think people study elephants very much, because it's pretty difficult to keep the adults in a healthy, you need a zoo to study elephants. So that is an elephant. What about this one?
Snake.
Yeah, this is a snake. And snakes develop kind of in a coiled shape. It's quite crazy. You can see all these little segmented patterns along the snake body, and those are those are the rudiments of its vertebrae and the spinal cord. So how the spinal cord actually develops. It's a timed process, and it occurs in intervals. So the intervals between when the snake vertebrae are segmented are much shorter and faster than humans, but it takes the same amount of time, which is very interesting. So yeah, that's a snake. I'm pretty sure that's the last one. Yes, it is. Okay, and these are some images I took last week, actually, of zebrafish embryos. So this is after I've fixed them in paraformaldehyde. So they are a little busted up in the tail and the yolk here, but this is the head, and this is the spine and the tail. And these are actually what we call WISH, or whole mount in situ hybridization, which is a certain type of experiment that I'll get into later. But essentially what it shows is where a gene is turned on in the fish. So normally these would be transparent, but you can see a dark staining, and that's where a gene is turned on in the fish. So it actually visualizes where a gene is active, which is really important, because when you know where a gene is active, you can know maybe how the gene works or how the gene functions. For example, a gene in the tail would never be involved in brain development, but a gene that you see in the brain, you could see where it's expressed, and you're like, okay, maybe it's involved in forming the brain. So yeah, we see this gene is in the head and along the vasculature of the tail. So this gene is actually a flow sensing gene. It's called KLF2A, and it gets turned on when blood flow in the blood vessel is high. So wherever you see sort of, well, it also is turned on in the development of some organs. So these are the viscera. These are some of the organs of the embryo developing. Same with here. But this faint right here, this faint staining, is actually the dorsal aorta. And that's where the gene is turned on in the fish. So I'd say right here as well, or dorsal aorta / cardinal vein is where it would be. So yeah, so this is actually a gene that senses blood flow. And as you can see, this is 2 days old, 3 days old, and 4 days old. So we're going from youngest to oldest at the bottom here. All right. So studying embryonic development is important because it allows us to understand the mechanisms responsible for complex biological processes such as cell movements, signaling and differentiation, tissue and organ formation, also known as organogenesis, as well as spatial and temporal genomic regulation of development. So like I showed you on the last slide, these genes get turned on and off in space and time within the embryo. And that actually, like I said on the first slide, that controls turning a single cell into a billion cells in the right way so that you get an animal in the end. So yeah, genes are turned on in location and time to sort of control development. This is a very interesting developmental process that I decided to include in here. So this is electron microscopy cross sections of a chicken embryo undergoing something called neuralation. And neuralation is the formation of a structure that is along sort of the back where the spine would be, called the neural tube. So the neural tube is, you can see it here. This is the neural tube after it's finished forming, but the neural tube is what develops into the spinal cord and the brain. Under here, we see what's called the notochord, and that structure controls development of the surrounding regions, including the neural tube. And then along the sides here are Semitic mesoderm, so the somites, which eventually form the vertebrae. So these somites will actually fuse around the neural tube to form the vertebrae in development. But to get that neural tube, we have this sort of process occurring where we get a flat surface and it bends in the middle due to these cells constricting at their apical sides. So we see cells kind of shorten to make from columnar shape to become triangular. And that creates sort of this curve along the back of the embryo. So right here, we see this is a medial hinge point, and these two folds come up together to converge at the top. And once they connect, they pinch off to form a tube, and that will form the spinal cord and the brain. When they pinch off, they form this other cell population called the neural crest, which is also very important. The neural crest is responsible for things like bone formation. It also forms a part of the heart. It's also important for pigmentation of the body. So when it pinches off, this cell population is formed, and that will actually provide your entire body with pigmentation. Albinism is associated with defects in migration of neural crest cells. So these cells right here, I guess, when it pinches off, they would be above here. These will migrate around the body to sort of provide melanocytes to the skin and pigment the body. So understanding how a tissue is generated provides clues as to how development can fail, and that results in disease that we see in maybe a newborn baby or an adult later on in life. So an example is Spina Bifida. So when this process goes awry, or when it goes wrong, the neural tube will fail to close at some certain point. So this will fail to form a full tube, and it will be open at some points. And that can result in what's called spina bifida. And here's an image of spina bifida. You can see the spinal cord is actually exposed on the back of this newborn because early on in development, this is in a chicken. I'm not sure when this would occur in human. I think around the 3rd week of development, so 21-ish days, this would occur in a human. And that later on, if it fails, would result in spina bifida when the baby is born. So knowing how these things go wrong can help us understand that things later on might result in disease. So we can, by knowing this, we can sort of develop clinical practices that can maybe help us circumvent things like this. I know right now for spina bifida, they're doing in utero surgeries when they find it in ultrasound. So they will actually do a surgery on the baby to fix this while it's still at the fetal stage in the uterus, which is crazy how they can do that. But maybe something in the future we can do is sounds very sci-fi, but we could genetically sample the baby. If we find issues that may result in neural tube defects, then we could genomically edit the baby to fix that in the genome before it results in the error. So depending on what you believe, there may be some ethical issues in there, but it's just theory at this point. It's just a thought experiment at this point. No one's actually done that. We can't do that at this point. So right now we're limited to surgery to fix an issue like this.
So
Okay.
Tarek, we cannot, sorry, can I ask a quick question? So we cannot do it because there are restrictions about it. Like for instance, can we do the CRISPR technology, gene editing? Like, so it's possible to edit, but we're not allowed to edit because of ethical issues.
Yeah, so currently with CRISPR, it's illegal to do genomic editing on humans. It's a criminal act, which makes total sense. CRISPR isn't mature enough yet to make sure that there would be no off-target consequences. With CRISPR, it targets a very specific sequence, but we're not entirely sure that when we use it, we won't accidentally damage some other region in the genome that would result in some other birth defect. So if that would happen, suddenly you think you're fixing a problem, but creating much more. So it is illegal. I did want to include talks about that in the presentation, but it was already at 40 slides, but we can talk about that after. There's actually a scientist who used CRISPR technology to cure or sort of create HIV immune newborns, and he's in jail right now because of it. But
and where
I'm
is
not
he
sure
from
if
again?
the newborns are normal. You know, I'm not sure. I think China. I think he's Chinese.
Okay.
Yeah, I'm almost positive that he's Chinese. I would have to check on that again. But yeah, he did use CRISPR gene editing on newborns. Sorry, on the embryos, which then developed into the newborns. And I'm pretty sure the babies are completely normal at this point, but of course it's illegal to do something that we have no idea what the consequences are. So he's in jail for it, which makes sense. You shouldn't be doing something like that, especially with science. We want to keep it pure, and we want to make sure we know what we're doing with our techniques.
You have a question, Khalid?
Yes. CRISPR. I didn't understand what is CRISPR.
Oh, sorry. So CRISPR. So CRISPR so let me go back to the beginning. CRISPR is actually a bacterial immune system that bacteria use to fight viral infection. Scientists have taken it out of the bacteria and used it as a genetic engineering method. So the genome is made up of sequence. We have letters in our genome. There's 4 letters that the genome is. I think it's a 6 billion long sort of string of these letters. With CRISPR, we can choose a sequence and replace it or repair it. And CRISPR is sort of our method to gene editing. Say a gene was destroyed in the genome, we could use CRISPR to replace the gene with the proper gene so that development can occur normally. So CRISPR is essentially a gene editing technology at this point.
So Tarek, would you say that 6 billion sequence, they're all combinations of these 4 amino acids, ATCG, all over, like just these 4 kinds, or they're different other kinds?
They're just those 4, yeah. I'm not sure if it's 6 billion or 6 trillion. I can't remember. Okay. It's a really big number, but yeah, it's ATCG, and there's just, I wish I had a picture of a sequence in this PowerPoint, but I don't. I can share maybe a picture later. It's just a very long sequence of ATCG, ATC, just combinations of ATCG, and that is what sort of corresponds to a protein sequence later on. I'll talk about it later more. I have that in the PowerPoint, but yeah, so if we control the sequence of a gene, we can control the gene's effect. That's pretty much what the idea is. And that's what CRISPR allows us to do. Other questions?
So just if I want to make it easier, like, I'm going to make it painfully simple. It's not that simple. So the professor who got the Nobel Prize for CRISPR, she talks like, oh, just think about it's a Word document, and you go in there, you find there's some grammatical mistakes, so you can go and actually fix it. So just like binary, pretty much the entire binary world is nothing but the combinations of 0 and 1. Our living world is a combination of those 4 ATCGs. So it's possible to see the sequence. And if we identify something is wrong, you can literally go just the way you use Photoshop or the softwares to fix photos and videos. I mean, it sounds sci-fi, but you can actually do it. I think that makes development biology so amazingly fascinating. And I also think that that's how development biology can also make us humble, not to be so proud about ourselves. That's what I was thinking when you're showing all the embryos, like, interesting. I can tell which one embryo is from which one, but we start pretty much from the same journey. Sorry, exactly. I'm enjoying the lecture so much. So go on.
No, yeah. There's a classical developmental biology sort of speaker. His name, I think, is Ernst Haeckel. Do you know who he is? I think he has some involvement in anthropology, but he had this quote that says, ontogeny, which is development or the process of development, recapitulates phylogeny. So that quote pretty much is saying that development is very interesting. If I go back to some of the previous pictures I have, it's very interesting how a cat can look like a snake or a lizard. And what that thinker was saying is that as an embryo develops, it sort of goes through the stages of evolution in the past. I'll show you some pictures of human embryos. Well, they're schematics, they're not actual human embryos, but yeah, it's quite fascinating how we can go from that form to a full organism, an animal with trillions of cells. Okay, so moving on, that was just a short intro. I'm going to go into some basic cell biology principles that will kind of help us understand the full picture of development. First things first, simple question. What is a cell? A cell is the most basic unit of life of which all living organisms are comprised. Organisms can be unicellular or multicellular. So an example of a unicellular organism is a bacteria. It is on its own a single cell, and it is also its own organism. Multicellular organisms are pretty much everything else that we can see and we know, like bugs, humans, snakes, horses, anything that has more than one cell. So here's some images of cells. So here are human erythrocytes, which are a fancy name for red blood cells. And this is what gives your blood the red color, pretty much. Each cell is shaped very uniquely. I'll go into different cell types, but you can see that these red blood cells compared to just a skin cell are very different looking. And that also depends on the genome, right? So this is a picture of human skin cells, and the yellowish is a protein called keratin, which is the same protein that's in your fingernails and hair. And I'm pretty sure the bright blue is actually the staining of the nucleus. So I think this one is, I didn't actually read it in the article they published this, but I think this one's undergoing a cell division. That's why you see sort of the separating nuclei. And this is just a nucleus. But yeah, okay, and here are some examples of organisms. So E. coli, this is real images of bacteria under a microscope. These are single-celled bacteria, E. coli are. Whereas humans have around 700 trillion cells. So you can be an individual living organism with one cell or 700 trillion cells, which is quite crazy. Okay, so on to general cell anatomy. So the very first thing about a cell is the cell membrane. It's the most basic thing that keeps the cell together. And it provides a barrier to allow for a controlled internal environment. Without a cell membrane, the cell would have no way of keeping its molecules and contents inside of itself. So it wouldn't be able to exist. So the cell membrane separates the inside of the cell from the outside so that the inside can perform chemical reactions and things that are required so that life can occur. The first of the organelles, I think I'm going inside out on this slide, is the nucleus. And the nucleus is super important. If you're going to remember one of them, remember the nucleus. It houses the genetic material in the form of DNA. So when I say genetic material, I mean what we were discussing earlier, the ATCG, the sequence that encodes the instructions to life. The endoplasmic reticulum, oh, sorry, on the diagram, this is the nucleus right here. Endoplasmic reticulum is the next thing out, and it is the site of protein synthesis, lipid synthesis. So it builds proteins as well as fats, and it also stores calcium, which is important in signaling, as well as, I know calcium is important in muscles as well to allow for muscle contraction. The next one is the Golgi apparatus, which sort of is the next organelle down the line from the endoplasmic reticulum. It is the site of protein modification and packaging. So the proteins are encoded in the nucleus. They're made in the endoplasmic reticulum, and then they're packaged in the Golgi to be shipped out of the cell, or to be moved to the membrane, or to be sent inside the cell to remain. Mitochondria, I think a lot of us are familiar. It's the site of energy generation. And cytoplasm is essentially the fluid that fills the cell. It's the dense liquid containing molecules required for metabolism and cellular processes. Okay, so cells are not just one type. There are many, many different types of cells, each having specific roles. Some examples include hematopoietic cells or hematopoietic cells. So these cells are essentially blood cells. So you can see from a single hematopoietic stem cell, which is a cell that produces all of the different kinds of cells. You can think of a stem cell as like a master cell that is able to make any cell type. A hematopoietic stem cell can create, if you look down the line, immune cells, so natural killer cells, T lymphocytes and B lymphocytes, which function in the immune system, so they respond to infection. And then down the myeloid progenitor lineage, we see if the genome directs it to go this way, we get red blood cells, we can get platelets, macrophages, which are also immune cells, eosinophils, basophils, and neutrophils. So there's a lot of cell types out of just one lineage, right? Another example are neural cells. So these are the cells that would be in your spinal cord, brain, peripheral nervous system. They're responsible for allowing you to feel and sense your environment, and also think, because they're in your brain. So some examples include neuroepithelial cells, which are the stem cells. Those turn into radial glial cells or ependymal cells. And then those turn into either neurons, oligodendrocytes, or astrocytes. So neurons are the functional cells in the nervous system. They sort of are the ones that carry the electrical impulses in your brain as well as your body. They sort of send signals to one another to sense your environment. Oligodendrocytes are supportive cells to those neurons. So they're known as like glial cells, if you've ever heard of those. They insulate the neurons to ensure that the signals are sent properly. Astrocytes are cells that are involved in forming the blood-brain barrier. And then this is a sort of a type of neural cell, but it comes from hematopoietic lineage. It forms a microglia, which is actually a type of cell that is involved in pruning neuronal synapses. So it'll sort of add and remove connections between neurons, which will help your brain develop and form connections between neurons, which will eventually change the way you think. Microglia also have been associated with Alzheimer's and dementia, because overactive microglia can prune your neural connections too much, and that can result in, later on in life, Alzheimer's and dementia, which is quite crazy. Some other cell types are epidermal or skin cells. Muscle cells, there's three types: skeletal muscle, which is connected to your bones, so like your bicep, your quadriceps. Cardiac muscle is the muscle in your heart. And smooth muscle cells surround your endothelia. So that would be the cells that allow your veins and vessels to contract to increase and decrease blood flow. Speaking of blood flow, we have vascular cells as well, also known as endothelial cells, and many others. So those are just a few. It would be an endless list if I could list them all. So another very fundamental thing that I wanted to show you is this hierarchy, that cells comprise tissues, which comprise organs, or organs are comprised of tissues, which are comprised of cells. So it's this important hierarchy because cells have to work together to form a tissue, and tissues have to work together to form a functional organ. With only one cell, you can't do much unless maybe you're bacteria and you are one cell. But in humans, a single cell can't do much. It has to work with other cells to form an organ system. Okay, yeah, so on to cellular function. So what cells actually do. So all cells undergo a variety of common functions. First one being metabolism, which are the chemical reactions that occur inside a cell to support life. An example of metabolism is when you work out, you feel a burn in your muscles. That's actually your cells making lactic acid because they're producing energy, and it's a byproduct of the energy production sort of process. Another common function is division, cell division. So this image is a division of mitosis. I'm pretty sure the top row just shows the genetic material, but here we see the chromosomes are sort of condensed, and they arrange themselves along an axis, and they pull apart. So we get replication of the DNA and then division so that each cell has an entire copy of the genome within them so that they're completely independent of one another now. So we get one cell producing two separate cells at the bottom here. Next, we have gene expression. So all cells express their genes except for a few types of cells lose their genome. Actually, I think red blood cells actually don't have a nucleus when they're mature, but that's very specific. Almost all cells express their genes, and through gene expression, they produce proteins. I'll explain that more later. And then another thing all cells do is apoptosis, which is programmed cell death. So cells will kill themselves in the case of maybe they're becoming too old, they become cancerous, so they have a program that makes them kill themselves before they form a tumor. Of course, when that program also gets damaged, then you get cancer, but apoptosis is the process of programmed cell death. I just want to draw attention to the cycle up top here. This is the cell cycle. So cells will often, once they divide through mitosis, they'll undergo a growth phase, which they undergo normal metabolism. They sort of prepare for replicating their DNA. Then they go through S phase, which is they replicate their DNA. And then they go through another growth phase where they continue their metabolism. They work to produce the proteins that they need to perform their function. So if this was a neuron, it would maybe be sending electrical signals. That would be during the growth phase. And then we see mitotic phase, where the cells have a doubled amount of DNA. So they're able to go through this mitosis, which will allow them to split apart and produce two new daughter cells. So that's the cell cycle that kind of encompasses all of these functions as well. Another term that I want to emphasize is gene expression. So I'm going to probably use that term a lot. That means the production of proteins that are encoded in the genome. So like we were talking about before, the genome is a sequence, and the sequence corresponds to proteins that are made from it. I could give a whole lecture on how that happens, but that would be too long. So just trust me that gene expression means we're making proteins that are encoded in the genome, and those proteins themselves have a function that they perform. Okay, this is really cool. This is what I wanted to talk about for the formation of the digits. So during embryonic development, we have sort of this webbing in between the digits of the hand. But as humans, as adults, of course, if you just look at your hand, there's no webbing between them. So how do we get rid of this tissue between the digits? The cells between the digits actually undergo apoptosis, which is that programmed cell death. So as you can see here, we're starting with this interdigital tissue. So the tissue between the digits. And these glowing cells here are cells that are undergoing that cell death program. So it extends all the way down into here, and they'll just die off on their own so that they can separate the digits into unique structures, which is very, very cool. I really thought this was interesting when I first saw it. So
I have a question.
You guys
Sorry.
think the same. Yeah, go ahead.
Digits. What do you mean by digits? Like the...
Yeah, so I just mean the fingers or the toes.
So it can be anything, like anything that's kind of like structured bones in the future?
Yeah, so I'm specifically talking about like the fingers on your hands and the toes on your feet. This is like specific. This is a mouse. So this is a mouse's front paw or hind paw. This occurs before bone forms, so you don't have to worry about the bone and everything. It's just really soft tissue here to begin with. It's just cells really, but the genetic program of the cells in between the digits will activate a programmed cell death, which will remove them so that you get distinct fingers. So at one point when you were an embryo, this happened.
So it's
With, like,
everything is
some.
kind of like, sorry,
No,
everything is
go
kind
on,
of,
Rista.
sorry, everything is kind of like coded. I don't know. I know I did some coding like in computer science. So everything has like a timeline, and according to the coding of it, it kind of like does what it's supposed to do. That's kind of what you're saying.
Exactly. Yeah, exactly. So this would always happen at a specific time point at this stage. So the genome of the cells in between the digits would tell the cells, okay, it's time to start this programmed cell death, so that in combination, at the end of this sort of process, we get distinct fingers forming once these cells in between the fingers die off. So it's sort of counterintuitive. Instead of you thinking, oh, fingers grow out from the palm, it's actually you have an entire webbed hand, and the cells in between will die off to form these distinct fingers.
And
So
I had
if I want
another
to say
question.
sorry,
Sorry.
go on, Rista.
I just wanted to ask about genome. What does, like, is it a process? Is it a cell? Is it a,
Oh,
what is
so when I
genome?
say genome, I mean the DNA. Right here, this slide. So the DNA is this double helical strand. There's a lot of DNA in the nucleus. So when I talked about it on this slide, yeah. So the nucleus houses the genetic material in the form of DNA. So you can see this is the nucleus, and inside a cell, and inside the nucleus is the DNA, which is the genome. The genome is DNA. The DNA is the physical form of the genome. Yeah. And the DNA itself encodes 4 letters that sort of form a sequence that can be read off to make a protein.
So is this when, I think in 2007 or 8, I think, who was it? I don't know, probably Bill Clinton, who there's this huge announcement that finally we have decoded the human genome sequence.
Yeah.
Okay.
Yeah, so yeah, that's exactly when I'm talking about the genome. There's this whole project to sequence. When we say we want to sequence something, that means we want to find out the order in which the letters occur. So yeah, in 2008, it was probably, I think it was like $1 billion program where they sequenced the human genome completely. I wish I could pull something up to show you, but there's online databases which just completely show the entire human genome sequence, and that is contained in the nucleus of the cell. So an important thing.
Let me ask you a very naive question, Tarek. I mean, I'm sorry if it is a very stupid question. So let's say,
no worries, yeah.
how a person looks like. Okay, this is the shape of that person's nose or eyes. So does it mean that the entire genome sequence of an individual is coded in each of the cell's DNA, or the gene gets expressed in a particular cell? For example, my eye color, it's one kind of gene expression. My hair color, it's one kind of gene expression. So where does it stay stored? Like, is in the embryonic stage where all of it is stored, and then the protein gets expressed when it is, okay, this is the hand, this is hair, this is eyes? How does it work?
So let's see. Okay, I'm about to hit that on this very next slide. So
okay,
this slide is the idea called the central dogma of molecular biology. So this is pretty much the basis by which molecular geneticists and molecular biologists think. This process is just always in our head, and we're always thinking about this process when we do experiments, when we do research. Exactly what you asked is this right here, this point. So all cell types have the same genome, but are different because different genes are on or off in each type. So let's think about, like you said, let's think about maybe a brain cell versus a cell on your skin. They both have the same genome, but they control which genes are on or off in order to make sure each cell has their unique identity. So I go on, I think right after this slide, I'm going to an example of what I mean by that.
Okay.
And then maybe if you are still wondering, I can explain it. But
okay.
Yeah, so this is the central dogma of molecular biology. This is a really, really big idea that occurs in molecular biology. So DNA is in the nucleus. It's that double helical classical structure that we see. And DNA stands for deoxyribonucleic acid. So it's a sort of molecule that contains a sequence. So it's a physical thing, but the physical thing is in a certain order that tells the cell what the sequence is. So DNA essentially serves as a long-term information storage molecule contained in the nucleus. And the double helical structure actually provides stability, so we don't get the DNA breaking down over time, because of course you want your DNA that encodes everything about your body to stay intact your whole life, right? So it's very, very stable. And because it's stable, we can't really move it. So we can't really take it and make a protein directly off of it. We need an intermediate that is sort of temporary. So we use RNA. So when we transcribe DNA, we form something known as messenger RNA, or mRNA, as you might have heard in COVID vaccines are actually mRNA vaccines. Same molecule. So RNA is called ribonucleic acid, and it is a temporary information molecule that carries the instructions to make a protein. So in the nucleus, the DNA would reside. The DNA would be in the nucleus of the cell in that structure that I showed you way back on this slide. So the DNA would be in here. RNA would be made from the DNA. It would be like someone copying down a sequence of letters. And then like maybe someone takes a piece of paper, looks at the DNA, copies down the sequence, and then they can move the piece of paper to somewhere else in the cell. That's what RNA does. RNA can copy the DNA's code and move out into the cell, whereas the DNA has to stay in the nucleus. So from RNA, it carries the instructions to make a protein, and that's in the sequence. So a protein is the molecule that performs a cellular function. It includes enzymes, receptors, ligands, structural proteins, storage proteins, transport proteins, and antibodies. I guess I'll go over these. Enzymes are proteins that catalyze a reaction. You can think of an enzyme as maybe one that I'll use in an example is actually like a salivary enzyme that breaks down sugar. That's an example. A receptor is a protein that can receive a signal. A ligand is a protein that's secreted to bind to a receptor to send a signal. Structural proteins, an example is keratin, so maybe your fingernails. Storage proteins are proteins that will function to store different molecules. Transport proteins are proteins that maybe bind a different protein, or they'll bind other molecules and move them around the body in the bloodstream or in a cell. And then antibodies are immune proteins. Okay, so yeah, so this is just a diagram overviewing this process. We have the DNA in the nucleus.
Sorry.
Oh yeah, go ahead.
For RNA, you said it's kind of like a photocopy of like a DNA. So is RNA like a human-made thing, or is it actually another cell within the human body that kind of just copies the DNA?
Okay, yeah. So RNA, that was an analogy. So RNA is a molecule, so it's a chemical made by the cell as well. It's naturally just made inside the cell. Yeah, if that makes sense. Yeah. And
that helps the process of cell division when they're like copying each other and making it like another.
So cell division is a bit different. We don't need to express any proteins during cell division, really. So this is cell division right here. In cell division, the DNA will just replicate itself. It doesn't need RNA. But once the cells divide and they're doing functions, they're doing processes, we have this thing called transcription, where we make the RNA, and you can think of it this way. This is an analogy. DNA is like the library, okay? It's the library where there's a bunch of books. RNA is like your notebook. You go to the library and you copy down word for word everything in a book, and then you take it home. A protein maybe is like the contents of the book. So it's what the book says, it's the ideas. You can kind of think of it that way. DNA is long-term information storage. RNA is just taking that information, moving it somewhere else in the cell, and the protein is what that information is used to make.
Okay.
Does that make sense?
Yeah, a little bit, but yeah, I don't know. Of course, I'm not studying the whole thing, but I can get a big idea. Yeah, definitely. Now it's clear.
Of course, as long as you can kind of have an idea, that's all I'm looking for.
Thank you.
I also just have a question to clarify about, so each cell has the same DNA information, right? Like each cell carries that genome?
Each cell, yeah, so each cell, so a human person has a specific genome, but every cell in their body will have the exact same genome as each other.
Right. And it's like different processes that affect each single cell will affect that DNA or that genome. Right?
So yeah, so the genome itself will affect different processes occurring in the cell. It's such a complex idea to have in one slide, I know, but we would be talking about this all day if it wasn't.
No, I just wanted to clarify. So it's just every cell is individual in what happens to it and what goes on, but it's all the same DNA in every single cell.
So let me go back to this slide. So let's say we have a lot of neurons, okay? What will go on in the neurons? Every single same cell of that type, every single neuron will be similar. They'll be expressing the similar genes will be on and similar genes will be off in order to essentially identify that cell as a neuron, and so that cell can function as a neuron. Compare that to a red blood cell. A red blood cell will have a completely different repertoire of genes that are on to make sure that it can be a red blood cell. But they both originally have the exact same genome, but genes are turned on or off.
I think it's really similar to like also binary coding, like 0, 1, 0, 1, whatever is on and off, that kind of tells you what number it is, you know? So I feel like I'm getting it now. And so I understand like the genome is kind of like the structure that has the capacity, that's the same capacity for every genome in every cell, but depending on what is on and off, it decides the effectiveness or the functionality of the what it is. Like, is it a neuron? Is it a, I don't know, is it a something like red blood cell or whatever? So yeah, I think I understand.
Exactly, yeah. Okay, I'm going to move on to an example just to clarify this even more if it's not clear already. So all cell types have the same genome, but are different because different genes are on or off in each type. So an example is, say we have these cells. So each of these is a cell, and this is going to be in the salivary gland. So this would be like in your mouth, where the cells produce the saliva. In the salivary glands, you would have this gene called AMY1, which is salivary amylase, which I mentioned before, is that enzyme that will break down sugar in your mouth. So that gene is expressed and it's secreted into the mouth. So you get this protein, produces salivary amylase in the mouth. Compare that to a neural cell in the brain. Do you think a neural cell in the brain would need to make something that functions in the mouth? It doesn't. So in the brain, the neural cell would have a specific function, right? It wouldn't need to make the same protein that the cells in the salivary gland do. So the AMY1 gene is present It's still in the genome, but it's turned off, it's silenced. The neuron says, no, I'm not gonna make salivary amylase because that would be a waste of energy. It would have no function in the brain. So no salivary amylase is produced. Hopefully that sort of clears it up. Okay, sounds good. All right, so that's
Can I
this
ask,
sort of.
sorry, Tarek, this is very interesting. I just want to ask one quick question. So that's how different genes are expressed. I mean, you know, on and off in different cells. But let's say Maddie and I, we both are humans, but we have different features. So why is that?
Yeah. So both of you would have, this would happen in both of you. This wouldn't change any of your features. But during development, when the face forms, the structure of the face, that is dependent on the genome, because cells need to express proteins to interact with each other, to move, to fold, to signal to each other. So because your genomes are different, the amount of protein your cells make, the time when your cells make the proteins, the location your cells will be when they make the proteins will all be different. So that will sort of change the way you look, the color your eyes are. Really, the color your eyes are is more dependent on sort of variants of a gene that you have, or of genes that you have. But it's sort of just small variations here and there. When they add up together, it makes a very big difference.
Okay, so which means that my gene pool is coming from my parents, which is different from Maddie's gene pool because they're different. If Maddie was my sister, in that case, what would it be that Maddie and I would have the same kinds of gene?
Yeah, so you would have much more similar variants of genes, as well as, so every human, you can think of it this way, every human has the exact same genes. No one has extra genes. The genes are always the same. This AMY1 gene is in every single human. But genes can be different, very different slightly. They can say a gene has a 1-letter difference from a different one in the sequence. That can change a tiny part of the protein, which will change how the protein works in each person. So you add up all the little differences in every single gene and every single protein. You have between two people who are unrelated, it's going to be a lot more than between two people whose genes came from the same parents, right?
Okay.
So it's the best way to explain it is that it's a lot of small differences in the genome that add up to make two very different looking people.
Okay. Okay. Thank you. Thank you. Thank you, Tarek.
Yeah. Okay, I'll move on. So model organisms is the next section I'm going to look at. So in developmental biology, this is kind of a funny image my professor used, but in developmental biology, we can't really use humans to study development. First of all, it would be unethical to have humans breed just so we could harvest their embryos. Obviously, that is totally illegal, totally not an option. I would never want to do that, nor would any other scientist. We do not condone that behavior at all. So we use model organisms. So the reason they're called models is because they're similar to us. We can look at them and wonder, okay, I wonder if that also happens in humans, because if it does, then we can relate it between us. So here are some examples. This one is a nematode. It's called C. elegans. So I think I can't remember the first name, but the genus species is C, like the letter C, elegans. This is your classic fruit fly. Everyone's heard of the fruit fly that you use for studying. This is Drosophila Melanogaster. That's the species name. This is a frog that has been used. Frogs, I'm pretty sure, were the first organism to be cloned, this species specifically, and it's called Xenopus laevis. This is the model organism that I study. It's called a zebrafish, or Danio rario. here's the chicken. I like his cartoon diagram. And then mice is the last one. So studying development in model organisms helps us understand how our own development occurs. And how do we know that the same things happen in these models than happen in us? What's the point of studying them if they don't even happen in us? Well, we're more similar than you think to these model organisms. If you look at this phylogenetic tree, the distance between these branches actually is relatedness evolutionarily. So, of course, here's humans, Homo sapiens, and this mus musculus is actually mice. That's the species name for mice. And we're in the same neighborhood as them. They are the most similar model usually that you can get to study humans, other than maybe monkeys or chimpanzees, which are not commonly used as model organisms, but have in the past. I'm pretty sure a group of scientists in China have successfully cloned macaque monkeys, which is pretty crazy. But yeah, so this is mice here. Xenopus, so this frog here, is pretty closely related to us. We're both vertebrates. So that means we both have a spinal cord and we both have vertebrae, which means we both have similar body plan. So making that body plan during development is a similar process. And that's why studying more similar organisms to us is important to understand ourselves, because at the cellular level, everything becomes much more similar than at the macroscopic level. Here's zebrafish. They're a bit further off from us than frogs because, of course, they're fish. They don't have limbs. They don't have legs. They do have a vertebrae and spinal cord, though. Drosophila fruit flies are even further off, so they're less related. They branched off during evolution longer ago. So, of course, a fly compared to yourself is less related than a frog compared to yourself, because flies are invertebrates, so that means they don't have a spinal cord, they don't have as similar of a body plan. And then out of all of these, the nematode would be the least similar to us, because they are microscopic nematodes that they're invertebrates, they're very small, but they are still animals. So, and they're easy to sort of use. That's why people use them as model organisms. They're easy to breed, they're easy to maintain. So there are some advantages as to using them, even though they're less related. So yeah, we study the ones that are closer related to us. We wouldn't study a plant to understand ourselves, really, because plant development is very, very different from us. As you can see, they branched off a long, long time ago. I don't know how long this is. This is like 1 million years ago at this point, almost 1 million. So this is probably 1 million years ago we branched off from plants, whereas frogs is much less than that, and fish and flies. So we're all animals, and that's why we can use other animals to understand ourselves.
This is so cool. I mean, I wish I could teach this, I don't know, man, nationally. We would be a lot more humble, not so arrogant.
Yeah, it is very cool. It's very, seeing it from an objective point of view to sort of, it's very humbling, you're right, to see like, wow, humans really aren't that special. We're very actually similar on the cellular level to frogs and mice and fish, so we can use them.
And then wouldn't that be an amazing world, actually understanding that we are so close to the other species? In that case, I mean, imagine that how much of the fate or some kind of, I don't know, superiority complex we harbor in us, men over, women. I mean, this is one of the things that I bring this in our discussion again and again, that you think that science does not have a politics behind it. You think that knowledge does not have politics behind it. Nothing is without politics. Understand it.
Exactly.
What do you think, Maddie?
Yeah, I totally agree. Everything has thought behind it and commotion and people talk, and yeah, everything is built on one another, right? We're more connected than we think, and that has an effect on things. So yeah, we can't
I mean, if this is how far we are away from plants, we are not very far away. I mean,
I think when you see it right in front of you, right? Yeah, no, definitely. It's fascinating for sure.
Yeah, thank you. Thank you so much. Interconnectedness. Yes, interconnectedness.
Yeah, and well, humans are cultural beings, whereas other animals aren't really. So unless you consider chimpanzees using sticks and tools as culture, humans are really the only confirmed cultural beings without argument. So I think the fact that we have culture and we talk, and there's always that aspect of culture in everything, even science, right? And especially because there's that culture, that also sort of helps the superiority complex kind of pervade, because we are cultural and we think we're better because we have culture. Whereas other organisms are left to adapt through evolution, whereas we can adapt through culture, which is maybe what makes it seem so much further of a gap to frogs than this, which is less than 1 million years difference. So maybe this isn't 1 million years. I'm not sure how long this is. I think that might be 1 billion, so maybe 700 million. That would make more sense. I think 1 million is too short. 0.7 of 1 billion. Okay, yeah. So yeah, it's still a very short time on the time scale of Earth, though, right? So Earth is what, 400 billion years old. So really, the difference is not that big. We're very similar to even just flies. Okay, so next is in vivo versus in vitro methods. So let me just define these. In vivo is Latin for in a living thing. So when I say an in vivo method, it means studying science using a living, breathing model organism, or an organism. So the model organisms that we just looked at are an example of an in vivo method. Usually in vivo methods provide more biologically relevant results, of course, because we're looking at a real biological system that is not changed whatsoever. The interactions between the cells will all be the same as they are in nature. But of course, it involves difficulties involved with working and maintaining living organisms. So obviously, if you have living organisms in your lab, you have to feed them. You have to make sure they're not in distress, because if they're stressed out, if they're hurt, if they're injured, if you're not treating them well, you're also going to mess with your results of your experiment, because a stressed animal is different from a healthy animal. So we have to maintain them. We have to make sure we treat them well. And that brings about ethical issues as well. Using living organisms, again, the only issue is it often introduces more experimental variability into the experiment you're using. So a living, breathing organism is more accurate biologically because it's in nature. It's how it was supposed to be, but it's also a lot more complex. So there's a lot of things you may miss. There's a lot of interactions you may not consider, and that can affect your experiment. That can add a variability. So that's in contrast to in vitro methods. So in vitro is Latin for in glass. Why? Because it involves studying biology outside of a living organism, or ex vivo is another word for it, in a test tube or petri dish. So in glass, because we use glass petri dishes, glass test tubes. So it's less biologically relevant because we don't have the complexity of the organism. It's not in nature. We're studying cells in a culture, right? We're studying cells in a dish, or studying organs in a dish, or whatever, right? So but the advantage is that it's usually less costly, higher throughput. So that means you can look at a lot more samples a lot easier and much quicker than you could with a living organism. And you can replace the use of animals for ethical reasons. That's another advantage. If you can use in vitro to prevent, to sort of circumvent the use of animals for laboratory studies, you can replace them for ethical reasons, which is another advantage to it. And then you can also better control for variability, because of course you're controlling everything that goes in or out of the test tube. So you can make sure that it's in a sterile environment, you control the interactions, you control everything. So that's another good thing. But again, it's less biologically relevant. So although you can control for variability, the results may not be relevant. So that's the one issue to in vitro. Here's a very interesting example of an in vitro model. So we call these organoids, which are miniature organs that are induced to develop autonomously from stem cells in a petri dish. So what I mean by that is you add stem cells, which are cells that can develop into any cell type, into a petri dish, and you add protein signals or chemical signals to tell the stem cells to differentiate into an entire organ in a petri dish from one cell, which is crazy. Here's an example of an intestinal organoid. So this is intestinal tissue, human intestinal tissue that was not taken out of a human. This arose from a single stem cell that they put into the petri dish and added signals to form an intestinal organ. Here's a brain organoid. So they actually got a part of a brain to organize on its own in a petri dish from a single cell. And that is just crazy. I find that crazy because if you can do that, then who knows, in 100 years, 200 years, you could make a full-on brain and it would just be, I have no idea if that brain could function or could have a personality, which would be crazy. It's super sci-fi, but it's happening. Like, this is a synthetic brain made in a dish, and it's not just this is a paper that was published in a serious paper, right? So they prove that this is brain tissue, that it organizes with the same layers that the human brain does, in the same stages and in the same way. And it also expresses the same markers that a human brain does. So yeah, they get a brain to develop in a petri dish. Here is a hair follicle. Multiple hair follicles. So the hairs, this is the follicle, and the hairs would be extending inwards like this. So it's almost like it's a sort of a ball, and the hairs extend into here from the outside. So yeah, this is where the hair would grow out of, into here. And sort of the objective of this paper was to find a way to produce hair without transplanting it off of someone, so that maybe in the future, if you get this to a level where you can produce enough hair follicles, you could treat burn victims. You could burn victims who have lost their hair, or people who have skin conditions. You can replace their hair with real hair. They transplanted it onto a mouse, and it integrated and became vascularized with the mouse's vasculature. So it actually grows on a living organism when you transplant it onto it. So this was a really cool
Tarek, what's vascularized, please?
So the mouse's vasculature grew into it and supported it, if that makes sense. So
okay.
So the blood vessels supplied it with the nutrients and oxygen. Yeah. And this is, remember, all of these came from a single stem cell and grew in a petri dish outside of an organism. This didn't come from an animal. These came from a single cell. So it's quite crazy how you can use this in vitro model to create organs in a petri dish. This is actually a really good thing ethically too, because now we can study human organs without having to harvest human embryos or human people, right? We can study the organs and how human organs work rather than studying mouse organs and just assuming it's the same in humans. We can study real human organs without hurting anyone, without taking embryos, without removing anything. So it's a very, very good model that's pretty new. This one is from 2015. And I'm pretty sure this was actually the very first organoid that they ever got to develop. So 2015 is when these things became real. And this is a paper from 2021, 2018. So it's very recent innovation. Okay.
And Tarek, could you tell us that where do we get the stem cells from? I mean, I know but could you tell us a bit about it?
Yeah, so the stem cells themselves would have to, this is a whole other conversation, but they've actually found a way where you can take any cell, you can take a skin cell and make it, let me just go back to the slide. This slide. So this is a stem cell, and notice a stem cell can make all of these cell types, but these cell types cannot go back to make a stem cell. So one cell type is stuck being itself. It can't change into any other cell, but a stem cell can make all the other cell types. Well, these researchers found a way to reverse this. They got cells to go back to a stem cell. So to get human stem cells to use to make organoids, you take any kind of cell that you want. I think skin cells are the easiest because they're easy to sample. You take a skin cell, you add these protein factors to it, and it will reverse lineage to go to the stem cell, and then you have stem cells to do the organoid experiment with, which is crazy, right? So that's how you would get the stem cells. Otherwise, I think the only other way to get a stem cell would be actually to take it from a human embryo. So maybe a researcher would have gotten permission to sample stem cells from an aborted human embryo that was spontaneously naturally aborted. Never in the past has anyone gotten approved to harvest human embryos, so that wouldn't have ever occurred. But now we have a way to not only get human stem cells just from sampling someone's skin. You can take it from your forearm and reverse it to become a stem cell. You can now make any organ from that cell that is possible at this point. So it's quite crazy how advanced these studies have become. So going on kind of from talking about humans in research, animals in research, there are ethical issues involved in developmental biology. So using animals for research brings up a lot of ethical questions. First of all, I kind of want to talk about the legislature that controls ethics in Canada. I don't know too much about other countries, but I had to go through this for my research. I had to get approved by the ethics board. But research involving animals in Canada is regulated by the Canadian Council on Animal Care, or the CCAC, as the primary oversight group. So they control guidelines on general care, facility design, wildlife use. If you're studying maybe ecology, animals for ecology, you would have to look at wildlife use. Farm animal use, for people who are veterinarians, you need to get approved to use farm animals for research. Antibody production, as well as euthanasia, GMO use, and much more. So that's just a few of them. So the CCAC requires regular facility inspections to ensure that ethical standards are being met at each facility. So if you get approved by CCAC and then you go on and start doing something that you're not approved for, they'll find out because there's regular facility inspections occurring. And then there's also provincial and federal legislation to ensure animals are treated and used ethically. Alberta has one of the strongest legislations due to the Animal Protection Act. And then federal legislation involves the Criminal Code and Health of Animals Act to protect animals from abuse, neglect, and disease. So researchers are only allowed to experiment on animals in accordance with their animal use protocol, which defines what they are legally allowed to do. So I can't just go into my lab, take a fish and dissect it and do whatever I want with it. I can't take a fish, make embryos, and do whatever I want with them. I need to be approved, or else it's a criminal act. So you can only do with your animals what you're approved to do, and the only way you can get approval is if it's ethically okay to do. For example, anything that overtly brings distress to an animal or is abusive towards an animal will never get approved. So the only way you could do that would be illegally, which they inspect facilities for these things. So that would be committing a crime. So there's a lot of ethics behind it. We're not just taking animals and doing whatever we want with them. There's a lot of legislation. Something I didn't include here is human legislation. I know that it is illegal to do genetic experiments on human embryos themselves. I'm pretty sure it has to be the only legal way to work with humans is before a certain stage. And I'm pretty sure that stage is like early fertilization. So just after the sperm and egg fuse, because once it starts developing into an embryo, it becomes a real living organism rather than a single cell. So who knows if it develops neurons, it could feel things. Like a fetus can obviously feel things. They have a brain, they have neurons. So I'm not entirely, I think it's very complicated on how human developmental research works. So, and I think it's also very difficult to get approved on doing experiments on humans. So usually we just use animals. So, or we use human cells, which is okay to do because there's a lot of human cells that we can get. Like I mentioned with the stem cells, you can get human cells in a way that is not ethically wrong. So yeah, there's a lot of guidelines. So now I'm just going to move on to general development after looking at cell biology principles as well as model organisms. How does development actually occur? So I'm going to start with the stages of development. This is quick overview. We already looked at this. We begin with an adult to form a fertilized egg, which contains the genetic information from the mother and the father. That genetic information goes on to control development of the embryo in these few steps until birth. So I'm just going to go over those in these next slides. So the first step I would say would be gametogenesis, so producing the sperm and egg, but directly after that is fertilization. So with fertilization, it results in the fusion of the male and female genomes, creating what's known as a zygote, which is a fertilized egg that begins development. Something very important to note is that only one sperm and one egg combine. And this is crazy how this is possible because there's millions and millions of sperm to one egg. So how does the egg control only one sperm fertilizing it? And there's actually molecular mechanisms that control that. So within the egg, there are these things known as secretory granules, which line the very outside membrane. And once the sperm fully penetrates the secretory granule, the egg, this surface right here is called the Zona Pellucida. Once a sperm fully penetrates that, you can see these sperm are digging in to the outside membrane. Once the sperm fully penetrates, electrical signals actually occur to release these molecules that will repel any other sperm from fertilizing the egg. So that ensures that only one male and one female genome can combine in an egg, rather than maybe if 3 sperm combine at once. That's not possible because there is a block that exists. So the sperm, of course, is produced in the male and it has a long flagellum, which allows it to swim towards the egg. The egg is much, much larger than the sperm. You can see here, this is one sperm compared to one egg, and it contains all the cytoplasm for the sperm, for the embryo to start developing from. So this is all the fluid that the embryo needs to begin developing. And it has all the nutrients within this as well. The sperm has this cap known as the acrosome, which contains digestive enzymes that break down the outside so it can burrow into the egg to fuse with the egg, and then the genomes actually fuse together once it's fertilized. So that's that step. And in a human, if you ever want to know where this happens, it usually occurs in the first 1/3 of the fallopian tube or the oviduct away from the ovary. So just 1/3 of the distance down the oviduct from the ovary. I don't have a diagram of that here. I don't think I have a diagram of that at all, but that's where this occurs.
But
okay,
what about,
so next,
what about the twins, Tarek?
Oh, so twins. So twins, there's 2 types of twins. There's monozygotic twins and dizygotic twins. So when I say zygotic, I mean a fertilized egg. So a monozygotic twin would be twins that came from one fertilized egg. And how does that happen? those would be, by the way, those would be identical twins as monozygotic twins. And the reason they're identical is because the egg actually splits into two. So you have two separate entities now that develop on their own. Dizygotic twins are non-identical twins, and those form because two eggs are released at once in the female and both get fertilized.
Okay.
Yeah, that's how twins work. Okay, so the following step after fertilization is cleavage. And what cleavage is a process of rapid cell divisions that divide up the cytoplasm of the zygote, so the fluid is divided up, resulting in a multicellular embryo. In humans, the embryo at this stage, called a blastula, will implant into the uterine wall to begin gastrulation. So the reason I say in humans, they will implant into the uterine wall is because in frogs and fish, they develop outside the female. So there's no uterine wall to implant into. So this only happens in mammals because in mammals, there's internal, or sorry, not just mammals. I'm not really sure which animals this occurs in, but definitely all mammals this occurs in. The fertilized egg will implant into the uterine wall to begin developing. So here we see this is after, sorry, I'll start here. This is the zygote. So right after fertilization, we get cleavage. So essentially the egg splits into multiple cells, and this keeps going on until it's a few hundred cells to form a blastula. And this is just a hollow ball of cells, really, is the easiest way to say what this is. And around day 5 in humans, so this occurs around 24 hours. So this is happening as the egg migrates down the fallopian tube to the uterus. So by day 5, we have a full blastocyst here. And that begins to implant into the side of the uterus. Now, something very interesting to know is that mammals actually do hatch from an egg, but it's during this stage. So on this slide, you see there's this sort of shell the egg is contained in that's called the Zona Pellucida. And it's a very hard shell that is smooth. It doesn't interact with the walls of the uterus or the fallopian tube at all. It just moves along it in a very smooth fashion. But the blastula contained inside, so on this image, this mass is very sticky. It will stick to whatever it touches. So as the egg migrates along the fallopian tube, it is contained in this. But once it wants to implant, the blastocyst or the blastula has to hatch, sort of. It has to break out of this Zona Pellucida in order to implant into the uterine wall. So it's not really your traditional form of hatching from an egg, but it's similar. So it's kind of an interesting thing that happens that you never really have heard of before. I didn't hear of it until I learned it. So so essentially the blastula will implant into the uterine wall, and it will sort of form an environment that it can begin developing in. So it'll form the extra embryonic tissues, such as the chorion and the placenta. It'll form the embryonic portion of the placenta, and then of course the female will start developing a placenta around the egg, and that will supply blood and nutrients to the embryo to develop. So after that happens, once it implants, you can notice that we kind of have a different looking structure here. It's not just a hollow ball of cells. Actually, sorry, I have a slide just before that right here. Okay, so this is more specific on the blastula stage. So right here, this is the blastula stage, or right here. You can kind of see it has this weird mass protruding from the inside. So it's still hollow, but it has a mass, which we call the inner cell mass. So the blastula is a hollow ball of cells that differentiates into regions that will form different parts of the embryo. The inner cell mass is highlighted in orange here for a gene that functions to keep these cells in a state where they can form the body of the embryo. So this is a gene called OCT4, and these cells are orange because they're stained for that gene. So the inner cell mass, also known as the epiblast, forms the embryo body. The hypoblast is this sort of ridge here, protruding into the cavity of the blastula, and that's what forms the yolk sac of the embryo. And then lastly, this surrounding region is what's known as the trophectoderm, and that will form the chorion and the embryonic portion of the placenta. So that looks like this kind of area around it where it interacts with the female uterine wall is what this trophectoderm will form. So every part of the blastula forms a different sort of thing at the end of implantation, but the inner cell mass is what we're concerned with. That's what the embryo comes from. Okay, so from that inner cell mass, we get this process called gastrulation. I know there's this funny quote that this one scientific thinker said. He said, the most important day of your life is not when you get married and not when you die, it's when you undergo gastrulation, which I thought was kind of funny because it is very important. So once implanted, the blastula will undergo gastrulation, which involves the movement of cells from the exterior into the interior of the embryo. So this occurs during week 3 in humans. So three weeks after fertilization happens, we get these cell movements occurring, which will form the body of which will sort of more define the parts of the embryo. So at this stage in humans, the embryo forms what's known as a trilaminar disc that is made up of 3 germ layers, the ectoderm, mesoderm, and endoderm. So I'll talk about those on the next slide. What I want to draw attention to you guys at first, though, is right here. This is sort of a diagram. I think this is a chicken embryo, but it's very similar to what happens in humans. It's almost identical. Actually, maybe this is not a chicken embryo. I can't remember. I thought it was, but it may be human. But we have this sort of slit that runs along the entire edge of the embryo, and the slit sort of extends. It extends this way over time, and as it extends, we get cells moving in through the slit into the interior of the embryo. As you can see, these blue cells sort of migrate, they migrate in and become these red cells, and then they'll sort of settle on the bottom, but some will stay in the middle and some of the blue cells will remain. So it's sort of what we call a bilaminar disc at this stage. So bilaminar meaning it's a disc that has 2 layers. Right here, you can see this is a disc with 2 layers. And then it becomes a trilaminar disc of 3 layers at 16 days. So 1, 2, 3 layers. And each of those layers are called germ layers. And those germ layers are important in forming specific organs and regions of the embryo. So this is where I'm going to talk about the 3 germ layers. So there are essentially 3 defined populations of cell layers that will go on to form the various organs and systems of the human body, as well as every other animal body during organogenesis. The first is the ectoderm. So the ectoderm on this last slide is the blue layer, and it's the layer that remains after the cells have migrated into the embryo. So it forms the epidermis, so the skin, hairs, I guess I said skin here, but epidermis is the same. Nails, the epithelium of the nose and mouth, so olfactory and mouth epithelium, the lens, so in your eye, it's the clear part that's underlying your pupil, the cornea, the nervous system tissues such as the brain, spinal cord, retina, motor neurons, facial bone and cartilage, pigment cells, and glial cells. That all comes from this blue layer on the very outside. The mesoderm, so the middle red layer, forms the kidney, gonads, so those would be the testicles and ovaries, the vertebrae, so bones, ribs, endothelial cells, which are the same as blood vessels, tendons, skeletal muscle, as well as the dermis, which is the layer that just underlies your skin. And then lastly, we have the endoderm, which is the yellow layer here, which will form the digestive and respiratory tract lining, urethra, bladder, liver, pancreas, prostate, and thymus. So you're probably wondering how a disc becomes a body, and that's because the disc actually folds this way inwards and it creates it's a disc, but it folds inwards to create a cavity. And that is during the process of organogenesis. So organogenesis is the process of organ formation. In humans, it begins during the 4th week. So the embryo folds from a disc into a 3-dimensional structure. During this time, the heart starts beating at 4 weeks, and the neural tube and many organs begin to form. So we can see here at day 22, it's in a disc shape here. This is sort of just beginning the transfer into a 3-dimensional structure. And we see the neural tube forming along here. That's at the beginning when I showed Spina Bifida. This process occurs here at day 22. And we get somites forming, which are the structures that create the vertebrae. And yeah, so this is sort of the disc shape. And just 6 days later, we get a 3D shape that looks sort of like a lizard, but this is an actual image of a human embryo. This is a human embryo here. So yeah, I guess this is the only image I've found to put in here of a human embryo. It was kind of difficult, but this is day 28 of this human embryo. So just less than a month of development, and we have something that looks kind of like a lizard. But in this diagram, we can see some labeling. So we have the brain is forming, the lens placode, which is a thickening of the outer surface of the embryo that forms the lens of the eye, so that hard part beneath the pupil. We have the nasal placode, which is a thickening on the outer portion of the embryo that forms the olfactory bulb. So like the nerves that innervate the nose, which allow you to smell. We see the heart is forming just under the chin here. So interestingly, the heart forms near the chin, and then later on it'll move down to the chest. And the heart, yeah, we see the heart is beginning to form here. Then we have these things called pharyngeal arches, which are very similar to gills, but they eventually will form into different structures of the face. So this pharyngeal arch will form into the mandible, so the jaw, and then a lot of facial bones as well as skull bones for the other pharyngeal arches. And then the pharyngeal arches also form all of the inner ear bones. So the bones in your ear that allow you to hear things are formed from these little structures here. Here we see limb buds. So this is a limb starting to develop. Here's the umbilical cord. Mesonephric prominence is the formation of the mesonephric tubules, which is part of the kidney. Here's the lower limb bud. So this is the arm, this is the leg. So you can see in relation to the heart is by the chin if this is where the arm is. But the heart, again, it moves down later on. And then we see sort of a tail at this stage of the human development, which will later on the legs and the body will extend sort of to leave this within the axis of the human, and we no longer have a tail at that point.
Tarek, doesn't this diagram help us to understand evolution really well in this very small amount of time?
Yes, this is what I was saying with that evolutionary thinker that said that sort of as you develop, it kind of recapitulates how evolution, sort of how animals look during evolution. It's crazy how humans have a tail and look like a lizard at this point, right? But all of us were this at one point, so we can't really argue. We can see actual sizes here. So this is 2.5 millimeters in real space. So that's, I don't know, I changed the image ratio, so that's not actually 2.5 millimeters. But yeah, 4.5 millimeters at this stage. So it's pretty small, but there's a lot going on for a very small area. That's the thing about development. Everything is so small, but there's so much happening. It's crazy. All right, so that's organogenesis. So yeah, we get the formation of organs happening here. Okay, now my last section sort of is just looking at some experiments that we do to understand developmental biology. So far, I've sort of just been showing you what happens, but how do we understand what happens? So I'm sort of going to bring up, there's a lot of techniques. We could talk about this for like a month, but I'm going to bring up this fundamental technique known as whole mount in situ hybridization, or WISH. It's a long wordy name, but it's a type of nucleic acid hybridization. So that means you're attaching a probe to RNA is essentially what you're doing, where the cells of an embryo are scanned using a probe that detects RNA produced from a specific gene. So you've designed a probe that tells you, like I explained this earlier, it tells you where a gene is being expressed and where protein of that gene is being made. So when the mRNA is detected, a stain is added that the probe can process to produce a dark color. And that is what tells us where a gene is expressed at the time point that we choose. So knowing where and when a gene is expressed can indicate what the gene's function is. So a gene can only function where it is actively being expressed. It can never function somewhere where it's not being expressed. So knowing where a gene is can tell us, hey, maybe there's a gene expressed in the hand that has something to do with, that means the gene has something to do with hand development. Whereas it wouldn't make sense if you said, oh, there's a gene in the hand, therefore it functions in the brain, right? It has to function where it is expressed. And it also functions at the time point that it is expressed. So genes can be turned on and off, but they're not permanently turned on and off. It changes when a gene is turned on or off. So here we can see, we can also remove a gene's function by destroying its genomic sequence, and that's also called a knockout. And that will allow us to observe development essentially in the absence of a gene, which will indicate its function. So let's say, let's bring up an easy example. Let's say there's one gene that controls the color of your hair, maybe. I'm sure there's many genes that do, but let's say there's one gene. Say I knock it out and suddenly your hair all becomes unpigmented because the gene can't control the color of your hair anymore. So destroying a gene sequence will essentially destroy its function. It will cause it to, it will make it so that the gene does not work anymore the way it's supposed to. So we can use this to do experiments to know how a gene works. So the example gene that I'm going to use, this is pretty funny, but biologists think they're funny, and they decided to name a gene Sonic Hedgehog. And there's a lot of history on that, but yeah, there's a gene called Sonic Hedgehog, and this is the in situ hybridization for Sonic Hedgehog. It is expressed at the posterior portion of the limb bud, so that's where your pinky would be. It's expressed where your pinky would be, and it actually functions to control the polarity of your hand. So it makes sure that your pinky is here and your thumb is here. And if Sonic Hedgehog doesn't work, then you'll actually form a pinky here, two thumbs in the middle, and a pinky here. It's really weird. It controls how your hand develops. I'm pretty sure it's that way. It might be the opposite direction, but it makes sure you have 5 digits that are unique. So again, I'm just showing a whole mount in situ. This shows where Sonic Hedgehog is expressed in a mouse embryo. So we see the blue here is wherever Sonic Hedgehog is expressed. So it's in the brain, in the forebrain, hindbrain, and then along the notochord, which is sort of underneath the spinal cord where the spinal cord forms. And then also this is the same image as the last one. This is at the sort of pinky of your hand in the limb bud as it develops. So it reveals it's expressed along the entire length of the embryo and in the head. So knowing that, when we destroy Sonic Hedgehog's function, we can see what an embryo looks like without Sonic Hedgehog. These are the same stage. This is a normal embryo. This is a Sonic Hedgehog knockout. So you can see this is the head, this is the eye, this is the tail, here's the hands. Sonic Hedgehog, the head looks like this with this long proboscis type thing. And then we see it's kind of transparent in the back, and then the limbs are very small. It only has hind limbs, actually. So this is because Sonic Hedgehog functions in controlling development of the eyes and the face. So when Sonic Hedgehog is destroyed, we get this proboscis structure, which is actually the nasal structures that failed to develop. And often these embryos have one eye. So they have a single eye, and that's known as cyclopia, that condition. We also see this transparent part is actually a lack of a spinal column. So without Sonic Hedgehog, the embryos don't get a spinal cord. Sonic Hedgehog is required to form a spinal cord, this gene. And then we see there's hands and limbs for this embryo, but there's none for this. So it's required for the formation of hands. So yeah, there's a few things that Sonic Hedgehog is important for. So this is my last slide. Just connecting Sonic Hedgehog to the clinical importance of knowing developmental gene function. So, as I mentioned, Sonic Hedgehog is important in separating the eyes into two separate fields. It makes one eye go into two. If Sonic Hedgehog doesn't work, you get cyclopia, so one eye. Human individuals with errors in Sonic Hedgehog gene have a disease known as holoprosencephaly or cyclopia. Disclaimer, this image might be disturbing to some people because there are humans in this image with issues in Sonic Hedgehog. So this gene functions in controlling eye field development. So errors in controlling that will cause eyes to be too close together or too far apart in some cases. Some cases are much more severe than others. This boy here looks normal, whereas this is probably the most severe cyclopia you'll ever see. So we have a single eye and a proboscis, just like this mouse here has a proboscis. We have a single eye and a proboscis, which would be the nasal structures, but they failed to move down into where the nose should be. And here we have the eyes are close together and they have cleft lips. So there's a failure for the face to join at the center. And that's because this gene called Sonic Hedgehog that we know is expressed in the head and functions to separate the eyes is destroyed in these individuals. Their gene doesn't work. So they don't make that protein that is required for this. So we get this diseased sort of phenotype for these individuals. So it results in defects in eye field separation. We see some people with eyes too close together, eyes too far apart, and in severities in between. So essentially showing you this, I'm just saying that knowing the normal function of a gene in development allows us to understand why these individuals experience defects, which may allow us to find routes of treatment. Okay, so conclusions. Developmental biology explains how a single fertilized cell can develop into an entire organism. Understanding the cell biology behind development can help us understand how things can go wrong, which may lead to clinical solutions to birth defects and development-related disease. And here's a nice image of a mouse embryo. I'm not sure what gene this is showing, but this is the expression of a certain gene that has expression in the veins, which is very nice to see. Any questions?
My question.
Mm-hmm.
That was really insightful. I'm like, yeah, a lot of things I didn't quite grasp, but I'm pretty sure I got the basic idea of the whole presentation, which was super interesting to know about these things that are out there. But when you say protein, and you know how we have like carbs and proteins that we eat, like, are they the same protein or?
Yeah, so they're this
protein?
Tarek, would you mind stop sharing the screen so that we all can see and we can?
Oh yeah, for sure.
Yeah.
Okay,
thank you.
perfect. Yeah, so protein. Oh, sorry, I forgot to just put on my acknowledgement. Sorry. Do you mind if I just reshare to put on my?
Yeah, sure.
acknowledgements.
Absolutely. Yes.
For the slides. So yeah, acknowledgements. Thank you to Dr. Cobb for permission to use CMMB 403 course content. And here's my references for a few of the things I had, but I didn't reference everything because just a few images and details were from those. So yeah, so all the other stuff was from Dr. Cobb and from my brain. So yeah. All right. So yeah, proteins. So these proteins are the same as what you talk about when you're talking about carbs, proteins, fats. It's the same thing, but when we talk about proteins in developmental biology, we're a lot less blunt with how we mean it. In molecular genetics, we mean it in a much more precise definition where a protein is a molecule that performs a specific cellular function. Whereas when we talk about protein in nutrition, it is the same molecules, but we just talk about them because we need to eat them, because in order to make protein, you need to eat protein, right? So when I say protein, sonic hedgehog, like what I was talking about, it's actually a signaling protein. So it gets secreted from a cell, it goes and binds a receptor on another cell, and it'll tell that cell to do something. So all of these proteins have specific functions, and that's what I mean when I say protein.
Thanks.
I mean, this question probably be a little bit sci-fi-ish, but what isn't sci-fi? So when you were discussing, a lot of the times you have mentioned that human embryos are not cloned, there are ethical issues, there are legal issues, and we know it. But if you look at history, let's say starting with Germany, so the whole point why we go through very rigorous research ethics board, we know that how in the German military have used human bodies for conducting despicable human experimentations, whether it is in Namibia or whether it is in Germany, we know that. But whether a government let's say in China, Canada does not have any prerogative what China does. We don't know all the weapons that are being developed in different parts of the world. We don't know, because these are intelligent information matter, so on and so forth. So do you think that it's possible that these things don't happen in university setups where there are possibilities of investigation, third parties, but if this is possible, I mean, obviously we know what is the consequence of nuclear weapon, but that's not stopping countries from producing it, right? So if it is possible to produce we know that it's possible. We know it's possible to produce human clones who don't have any parents, who don't have any social attachment, they're literally cloned. And when we have talked about gene editing technology, so you can literally edit gene. What happens to those individuals can be a lab case. Nobody's going to go on the street, holding placards saying, okay, you have done this, right? But it's possible that in future, hypothetically, theoretically, that just like biological weapon, some countries can decide to do this genetically enhanced human beings for warfare. Is that completely out of the realm of scientific possibility, or?
I think you have a point. I think CRISPR is not dialed in enough to be accurate enough to create a human that is a person that is a weapon because they're so enhanced. I don't think it's impossible. I think anything is possible in the future with this kind of field, but especially since people have cloned animals. There's been so many animals that's been cloned. So cloning is not out of the realm of possibility. It's completely possible. It's just a matter of us knowing which genes will enhance human function and maybe which genes will, if we change them, will completely cause us to. I'm thinking that if you clone a human and you change a lot of their genes, you can't really expect what to get until you find the right recipe of genes to change, right? You could have a mushy ball of tissue at the end of the cloning process because development is just aborted because you changed the wrong genes, right? So in the end, we're very far away because we don't even understand all the genes required yet for maybe height or strength or eye color even, or things like that. But it's completely possible, I think, to look at your child and to genetically screen your embryo, your child that's developing inside of you, and seeing, okay, this person has an issue in this gene, and we know that this will cause a disease later in life. We can genetically edit that so that later in life, they're totally fine. They won't have a disease. I think that is much more in the realm. I think changing so much about a person, I think eventually maybe, this is the issue, people have said designer babies. Have you ever heard about that term? Where,
yes.
Yeah, where people can, yeah, exactly, where people can edit their child's genome because they want them to be blonde with blue eyes or darker skin, less pale skin, things like that. It's not out of the realm of possibilities, but at this point, I don't think we know all the genes involved to do it anyway. And also CRISPR is not, like I said, accurate enough to make sure it happens without any off-target effects.
And in terms of, so another factor, I'm just gonna joining some dots here. So we have Neuralink, Elon Musk's project, that brain-computer interface. So we have talked about, you have talked a lot about how these, at a cell level, what happens. Now, in a brain, if a computer is constantly tracing every single neural firing, we don't understand it yet, but enough data is connected so you can understand that what kind of brain map produces what kind of emotion. So which means that in the near future, after collecting, let's say, X amount of data, we can possibly do, I mean, the whole computer design, like connectome, like how a computer is designed, understanding how the brain function works. And I mean, so there is a TV, I don't know if it is in Netflix or where, it's called Altered Carbon.
Okay.
So basically, the brain-computer interface is tapping on brain neural firing. And if it collects data at a certain amount of time, if it can actually clone your consciousness, and if the body can be cloned, which means that we are heading towards a theoretically possible, for example, when Alan Turing was talking about the possibility of a thinking machine, it was laughable, but it hasn't taken very long to actually see where we're standing at the moment in terms of artificial intelligence.
Everyone,
behavior, right?
Everyone has one of Alan Turing's machines in their pocket, right? Like
exactly.
Yeah.
Exactly. And my research being in the digital realm, one of the things that I constantly worry is that this exponential technology has a profit-driven logic that it will do anything necessary as long as it generates profit. Forget about ethics, forget about what it does to humanity. And if you look at the history, human history, modern history, or let's say 700 years of history, somehow profit has taken precedence over ethics. So in this realm, do you think that even from the stage, like let's say Jimam, Alif, they're in high school, they need to start thinking about the ethical implications? Jimam, what do you think about it?
I think, yeah, I think it's good to know about this kind of technology, that it exists, that Elon Musk is working on literally uploading your brain into a computer. This is his goal, right? With the Neuralink. At least I thought that's what I've read. I think it's important to know the technology isn't there yet, but we had flip phones like 10 years ago, and now we have smartphones that are as powerful as computers. Like, it's exponential, like you said, and anything is possible. especially with biotechnologies becoming involved, who knows what you could integrate together with biology. Really, if you think about it, biology is just a very complex physics system, right? It's on the surface, biology seems messy, but at the molecular level, it's very predictable. It's very, very predictable, and computers like predictable things. So I think engineering these kind of things are very real. I think it is completely possible. And in terms of ethics, yeah, we should be aware, especially if you start working in the field of science and research, in engineering, in physics, in math, in biology, you should know about all of these. It's very, very useful to know where you're at in terms of your ethics, what you're willing to do for your research, and of course, knowing what's available to you, what's at your disposal to do your research. So I think absolutely ethical questions should be asked.
And one other thing is that if you look at, and Jimam, I want you to give your opinion on that as well. So if we try to understand the integration of 3 different technologies, you have talked about biotechnology, and we have also talked about computational technology, particularly with the rise of cloud computing. It doesn't matter wherever you are. And with the emergence of big data, there are all these devices that can collect data. And then you have financial technology. The reason I bring financial technology is that in the past, whenever we have used money, we had some kind of materiality. We had either money printed based on gold standard or based on foreign currency standard. But as the money is becoming more and more digital, it means money is being created completely out of thin air. You and I do not have the capacity to actually check, understand what are the fiduciary policies behind money. So you have biotechnology, you have financial technology, and you have computer technology, and they march together. But what is the underlying logic that actually dictates the integration of these 3 different technologies? Now you have Vandana Shiva, a very prominent scholar from India, who also happened to have graduated from the university where I'm doing my PhD. So she has written a book, very powerful book. It's called Oneness versus 1%. So she's expressing her concern that you have right now, Bill Gates is the biggest landowner in the United States. It's not a particular farm company. You have these technology companies who have enormous projects in India, in places where you have lots of biodiversity, lots of ecological diversity, right? So what's happening, they're extracting all kinds of genetic information, which they call DSI, digital sequence information. Mm-hmm. So in the past, you would actually have to do all kind of genetic experiment to understand which breed of seed is compatible to a particular weather, and hence you can claim patent right. But now you don't even have to do that. You have an algorithm, you have this DSI, and when they extract the data from this in ecological or this life form, the ownership of the data is no longer of the people. You have, let's say I'm brushing my teeth, or whatever, any point of the data that you can extract from me. Extracting my genetic sequence is not that hard. When I say I don't essentially mean only I mean all forms of life, all forms of life. So Vandana Shiva's concern is that when all forms of life starts getting coded, starts getting dispossessed from, if you are taking my genetic sequence, it's my data, it's my life. But when you put a pacemaker in my heart, that data my heart produces is no longer my data. It's a proprietary data, right? So in a situation like this, young people who are sitting here right now, they need to understand the convergence of biotechnology, the financial technology, and computer technology, and all of them are governed by one profit-making logic. Jimam, what do you think about it?
So you're asking me?
Yeah, that's because I always talk about technology stuff with you.
Can you please repeat the question?
Okay, so I will ask
I'm
first.
sorry to interrupt. After
Go.
Jimam is done, I want to share my opinion.
Okay, sure. So the question is, if this computer technologies can extract all of your genetic data, I mean, when I say your, I don't essentially mean your life form, let's say the agricultural data, right? And using artificial intelligent model, they can come up with new variants of seed and they can claim patent right. But
Yes.
they could not develop the new variant of genetic sequence if they did not collect your variant, seed data, different life form data in the first place. But when you have a tractor that is collecting data from India, from Bangladesh, they're not asking your permission. They're just collecting data. You have cloud computing that is collecting data, right? So you have biotechnology, what Tarek has talked about, very brilliantly. Then we have computer technology, and then we have financial technology. But all of it is run to maximize profit. Does that make you concerned? What do you think about it? You think about technology, you think about artificial intelligence, you and I talk about it a lot. What do you think as a young man living in Bangladesh?
Okay, so I want to say about the Neuralink you just said.
Mm-hmm.
So Elon Musk is developing Neuralink. So
Mm-hmm.
if Neuralink is put it in our mind,
Mm-hmm.
then he can extract any type of data, what we are thinking, and he can control anything what we are doing. He just have to press a button, or he just have to give a command, and we will follow his command, and we will do anything he want. So he can control us with this Neuralink, and he can extract any type of data. He can get lots of data from this Neuralink.
Thank you.
So I
You,
think
Jimmy.
it will be harmful. And you talked about digital money.
Mm-hmm.
If you don't put Neuralink in you won't be able to get money. So you have to put Neuralink in yourself. You don't have any other option. You won't be able to buy food if you don't put Neuralink in yourself. So you have to it's you have to put it, and you have to give data, and Elon Musk will control you. You don't have any other option.
So basically, Elon Musk or anybody who has the power, capitalistic power, they will have the control over, like, for example, whether I give my data. Poor people don't have the rights. They don't have, they cannot say, okay, you cannot take my data. I mean, Rumi, what's your concern?
So first of all, it was really very insightful. I learned a lot from Tarek Bhaiya. I had studied this in my undergraduate courses, and I had to know a lot of things. A lot of questions is cleared now. Secondly, the thing that you're talking about now, which Tarek Bhai also said, designer babies. One of my professor in my university talked about it. The rich people can meet doctors or scientists and tell them what kind of babies they want. Secondly, the entire thing, the thing that you're talking right now, that is, this Neuralink and there's all this biotechnology and this computer and this finance, all these things will not bring good. Why? Because uh, firstly, the people, most of the people of the world, that major people of this world are poor. They don't have the access to all these facilities you're talking about, and that's why only very smaller a portion of the people have this facility, who can work in all who can work with computer skills, who can work with monetary advanced opportunities, and also biotechnology. So those people are profit minded. The only thing that they want is maximizing profit. So they don't even care. They will not care about anything. They will only think about, okay, I don't even care about invading anyone's personal land and take the information from them. I only want to maximize their profit at any cost.
that has been the history so far. I mean, whenever they wanted to extract profit, or continues to be, right? Yeah, so I mean, this is super fascinating discussion. I think we should continue having dialogue with Tarek, and I mean, there are so many things that I have questions, but obviously no one. We will probably. But Tarek, thank you very much. Honestly, it was mind-boggling. At the same time, this is what actually excites imagination among our youngsters. And these are the kind of conversations that don't go to the schools in the villages of Bangladesh. But I'm super excited that they are participating, they're asking questions. Alif, do you have any question to ask? Anything to contribute?
No, there is no question, as Tarek Bhai just made us clear enough. And if I have question, I will just watch the video if it is uploaded in the YouTube, and then I will ask you or Tarek Bhai.
But what's your you being a Bangladeshi young man, what are some of your aspirations, or optimism or concerns when you think about what we just have talked about?
About technology.
About developmental biology, and the other things that we have talked about so far.
In Bangladesh, the young generation like us have the dreams to become a doctor or engineer. But I think biology is an interesting subject. I didn't find any interest in biology before, but I find biology much interesting today due to Tarek Bhai. And I know about, I just have some knowledge about cell, and it was neuron from my school, but not enough, as Tarek Bhai said.
Wonderful, thank you, Alif. And Oyan, what's your opinion? What's your understanding so far?
I think there are some problems with development of biology, but there are some few. These are only few. We have huge, and that can contribute in our future hugely. In our life, they can prevent any of our disease before our birth. I mean, by watching our DNA and many things on. And I want to thank Tarek for this amazing conversation.
So someday Tarek will design, he will sit down behind the computer and he's going to write a bunch of code to decide, oh, so the next, I don't know, frog is going to be of this quality. He's going to write a bunch of codes and it's going to come out like that. What do you think, Tarek?
It's completely possible. I mean, there's so much meshing of all the fields, right? Like, there's especially now that biophysics, biomechanics, there's so much, like bioengineering is becoming huge now. Biotechnology companies are doing well and everything, especially with COVID. They've been able to shift their manufacturing to sort of the needs of COVID. But I mean, in terms of development and all of these interesting questions, it's crazy how meshed all the fields of science have become. And I mean, it's completely possible. Who knows, maybe in the future there will be some sort of machine where they take these the technology they've begun to develop on these organoids, the organs in the petri dishes. Maybe you're like, okay, I want a heart for transplant. Push a button and you get a heart in a petri dish. It's completely possible. They've been able to make brain tissue in a petri dish, which if you ask someone that in, I don't know, maybe if you said that to someone in the 1950s, first of all, they would have been like, I have no idea what you're talking about. And second of all, there's no way, right? It's, yeah, it's quite crazy how far it's come.
Tarek, I want to ask you this one question, last question to you, is that, so a lot of us, we don't understand that we have been able to push, I mean, the world has been able to produce a vaccine in record amount of time, in the short, small amount of time. And there, it wouldn't have been possible without actually biostatistics, biocomputation, how the artificial intelligence has been used to understand the gene sequence of the virus and to understand its behavior and how. So could you speak to that a little bit so that, I mean, we heavily talk a lot about technology so that they understand, oh, where is the technology, artificial intelligence is coming in the fields of, developmental biology.
Yeah, so a lot of the technology in developmental biology is really, other than experiments, a lot of it is very, especially with gene sequences, it's open access on the internet. There's databases of not only sequenced human genomes, there's sequenced mouse genomes, sequenced zebrafish genomes, every genome where there's a model organism. You can search up the gene you want, you look at it, you get your sequence, and you can work with it, right? So there's been huge advances, and especially for something as big, yeah, it is scary, I think, knowing the sequence of every living organism, but it does make it easy for research. We don't have to find it out ourselves. We can just type in the gene we want. But in terms of COVID, yeah, I think it has helped advance, especially getting the vaccines out as fast as they could. I'm sure the sequence of the mRNA vaccine was one of the first things they were figuring out. It's just so quick to work with sequence now that we have these powerful computers who can compute millions of lines of sequence for DNA, right? So I'm not sure how big the sequence is for the mRNA, the Pfizer mRNA vaccine, but knowing how effective it is, right? Of course, there's so many tests you have to do with the vaccine before you get it out to the public. There's animal testing. So, I mean, the right mRNA, you have to get the right mRNA, and that mRNA corresponds to a viral protein. Which viral protein do you choose? Well, it has to be one of the receptor proteins on the virus, right? Because that's what interacts with your body, or what things like that, right? So working with sequence has just become so much quicker and easier, and anyone on their laptop could do it in maybe 20 minutes. You could figure out a sequence of a gene and design an experiment using that. It's so much easier with computation now.
Yeah, I mean, at Guelph University, it's a huge, big university in agriculture and all kinds of veterinary, and and I know dozens of friends who are also doing their PhDs. You would feel like, so you are working in the field of, I mean, let's say you're experimenting with mouse or particular livestock, but then they're using so much computation. So as a teacher, I tell the students that, look, in the future, you become a biotechnologist, you become an anthropologist, you become a doctor, you become an architect. I mean, just if you did not know English, your knowledge door would be shut. You don't learn computer, you're gonna be in trouble. So, I mean, I tell this to my son, I tell this to students that, you gotta know this. I mean, computer is not essentially only for computer engineers per se.
Yeah, well, technology is a tool, and it's a very universal tool, right? It's applicable to almost every field, especially with the way research works. We gather so much data, we gather so much information that there has to be a way to sort it, to find information, to trade information, to work with information in terms of statistical tests and things like that. So it's a tool, and it's very important and useful.
Yeah, Rita?
I just wanted to ask a question. I know you have a Moroccan background, right? So I was going to say that how you said that it's a universal tool, it is, but some countries have access to it more than the other countries. For example, like when I was growing up, I was privileged and lucky enough to be the only person in the whole neighborhood with a computer. Not like everyone, and that was in the 90s. That wasn't even like 70s or 80s or anything like that. So you know, like I would ask you, like, have you, I don't know, like, is it similar in Morocco, or have you ever visited, or do you know?
Yeah, so I didn't grow up in Morocco, but I've been there, and you're right, it is similar, especially in the smaller towns. They're much larger families, so the focus of the family would be less on, I think the bigger the family you have, the general trend to me, I'm not an anthropologist in any shape or form, but from personal experience, my dad has a very large family, and 1 out of 6 of the kids went to university, and it's I think a lot of the focus is more on basic lifestyle needs if the larger the family you have, because there's more you need to do day to day to upkeep 8 people in one house, right? So it was definitely like that. I think I'm not sure about computer availability, but when I was there, there was computer cafes, and this was around like 2010, 2008. There were computer cafes available to like the neighborhood that people would go to, but I'm not familiar enough to comment on maybe how well versed they were with using technology.
Yeah, I mean, if you like, I'm gonna be probably I'll get a lot of heat for that. I mean, most schools in Bangladesh, when I say most, I mean, I have taught the most privileged students of Bangladesh. I have taught in the most elite private schools of Bangladesh. I used to be an economics teacher. So my students had access to all these technologies. I mean, me being a teacher for a long time, my family has access to all these technologies. But a lot of the students here, like for example, Oyun's dad, you know, I have called him and I have bugged him that please buy him a computer, or Alif's dad, or, you know, the thing is, living in Canada and working 3 years in technology companies, particularly exponential technology companies, doing my dissertation that is also connected to technology, I see how the power of technology, its societal impact, and how inequitable is the access. You know, this famous saying is that the future is already here, but it's not at all evenly distributed at all. It's not at all evenly distributed. So 99% students, more than 99% students of Bangladesh who are studying in public school. I mean, ask any of these kids here. So again, Jimam, do you think, you know, you have discussed technology in your school growing up, or, you know, or the places the way we discuss technology here?
Never.
They don't. We have started discussing that whether it is science, whether it is technology, it has the politics. And I define politics for them this, is that anything is developing, it benefits some people and it marginalizes others. So that you need to start questioning that who is it benefiting? So when you think about developmental biology, and when you think about genetic engineering, when you think about biotechnology, we do know that if one country wants to decide to develop a biological weapon, just design a deadly virus, design something. And we know that countries are building biological weapons. We know that. It's not a secret, you know? So this is where, Tarek, you can make a lot of contributions to many other places in discussing that, like, hey, what you said today, you know, you said it beautifully, that we are actually sophisticated physics. This was a beautiful way to say it, is that at a molecular level, you can literally understand, like you were saying, you know, and Rushda beautifully has pointed it out, that it's pretty much like programming, that which gene expresses, which gene doesn't, right? So anyway, I am super excited, and I really invite you, Tarek, consider joining us in future classes. And Maddie, please, your discussion was awesome. I'm waiting for next time Maddie's gonna say, okay, so on that day, I'm going to discuss this project, or you're working on this project, or you are doing this project. I mean, do come and drop by, whenever time permits you, okay?
Yeah, thank you.
I mean, you are invited 5 days a week.
Right.
Any day you are free, just, when it is Toronto time, it's 9 o'clock at night. I think it's 7 at your place. So Tarek, the invitation is open to you as well. Just, just drop by, just like Rusta does. Any day she's free, she just drops. And I mean, I want you to consider this as your extended family, like learn from them and also teach. So, exchange of knowledge.
Thank you. It's definitely a privilege to be even a part of this. So thank you for inviting us.
Pleasure. I will send you, I'll look you up, and I'll connect with you folks. Okay. So it was a pleasure. Everybody have a wonderful day in Bangladesh, and you folks have a wonderful evening in Canada. That's all for today. And Tarek, once it is done, I'll send you the link. Okay.
Okay. Sounds good. Thank you very much.
Thank you, Tarek. Thank you very much. Bye-bye. Rushda,
No
bye.
worries.
Bye, everybody.
Bye.
Bye-bye.